The overall goal of this procedure is to prepare the Drosophila flight motoneuron 5 for in situ patch clamp recordings. This is accomplished by first visualizing the Drosophila ventral nerve cord with the GFP-tagged flight motoneurons 1 to 5 through a 40x water-dipping lens. The second step is to use a broken, protease-filled patch pipette to apply protease across the GFP-tagged motoneurons in order to loosen the debris, glia, and trachea that obstruct the access to motoneuron 5. Next, debris, glia, and trachea are removed by gently sucking the loosened debris and glia into the pipette and pulling the trachea out of the way with the application of positive and negative pressure alternately. At this point, bright light is used to remove the remaining debris that cannot be seen with fluorescent light through a GFP filter. Ultimately, the cleaned motoneuron 5 can be seen faintly in bright light, but seen well using fluorescent light with a GFP filter. After rinsing the cells with saline for 15 to 20 minutes, they are prepared for subsequent in situ patch clamp recordings.
The procedure I'm going to show you today will help answer key questions in the field of neurophysiology, such as which ionic channels underlie which ionic currents, and in turn, which ionic currents underlie behavior.
Generally, individuals new to this technique will struggle because it is hard to see the subtle changes of the appearance of the neurons during the cleaning procedure, which is necessary prior to the patch clamp experiment.
A clean cell membrane is a crucial prerequisite for subsequent in situ patch clamp recordings, and since it